Good morning students. Myself Dr. Ragasudha, Head and Assistant Professor Department of Microbiology, Government City College is here with a topic on production of cheese and yogurt. What is cheese? A large part of milk produced is used in the manufacture of cheese. Cheese is the partially degraded concentrate of milk fat and casein with a small amount of milk fluid. The manufacture of all cheese depend upon the activity of microorganisms. Several hundreds of varieties of cheese are manufactured, and the amazing fact is that most of them can be made from the same batch of milk. This is possible by providing conditions that favor the development of selected types of microorganisms. Stated in another way, the quality and characteristics of cheese are determined by the biochemical activities of specific microorganisms. Cheese is one of the oldest human foods. Definition: cheese is defined as a consolidated curd of milk in which fat is entrapped by coagulated casein. Let us see some of the terms we come across in this particular video. Curd is solid clusters formed when milk separates into solids and liquids. Whey, the liquid part separated from the coagulation and curdling of milk. Casein, the protein found in milk and cheese. Rennin, also known as chymosin, it is an enzyme extracted from the stomach of calves. Rennet is commercial form of rennin used to curdle milk from cheese. Classification of cheese. They are classified into 4 types. The first type of cheese is based on moisture content. The first type of cheese is divided into 4 different types. Soft cheese, where the moisture content is from 50 to 80%. Example: cottage cheese, sour cream cheese, camembert cheese, and Ciota, feta cheese, etc. Semi-soft cheese consists of 39 to 50% of moisture content. Example: Roquefort, Danish Blue, Limburg, and Gouda cheese. Hard cheese consists of less than 39% of moisture. Example: cheddar and Emmental cheese. Very hard cheese consists of less than 34% of moisture. Example: Grana, Romano, and Parmesan. The next type of cheese is type 2, which is based on curd formation. It is of 2 types: acid curd cheese or cottage cheese, or rennet curd cheese. When acid is used for curdling of cheese, it is known as acid curd cheese. Example: cottage cheese and sour cream cheese. When rennet is used for curdling of cheese, it is known as camembert cheese or Limburger cheese. The third type of cheese is based on ripening. If ripening is followed by production of, in the process of production of cheese, it is known as ripened cheese. Example: Roquefort, Danish Blue, Limburger, and Gouda cheese. Sometimes cheese is not ripened. That cheese is known as unripened cheese. The cheese does not age. Is known as unripened cheese. Example: cottage cheese, sour cream cheese, quark, and soft cheese. The fourth type of cheese is based on mode of ripening. For the purpose of ripening, if we use bacteria, it is known as bacteria ripened cheese. Molds are used, then it is known as mold ripened cheese. Example for bacterial ripened cheese are Limburger, Brick, Gouda, and cheddar. And the microorganisms which are used for bacterial ripening are Lactococci, Lactobacilli, Leuconostoc, Propionibacteria, and Brevibacteria. The second type of cheese here is mold ripened cheese. Usually penicillin is used for ripening. Examples for this are Roquefort, Danish Blue, etc. In addition to these four types, there are a number of varieties of cheeses like Limburger cheese, where little salt is added to the curd. Linderkranj cheese, it is similar to Limburger cheese except considerable salt is added to the curd. Camembert cheese, the curd retains a much larger portion of the whey, and the microorganisms used for production of these are Geotrichium candidum and Penicillium camberti molds. In American cheese, the curd is precipitated after acidification with rennet. Whey is drained off. The curd is salted, pressed, stored for favorable flavor development. That is called American cheese. Whereas in Swiss cheese, fresh sweet milk is used. Streptococcus thermophilus is used as a starter culture. In Roquefort cheese, it is best known cheese made from sheep milk. Penicillium roqueforti is the starter culture. Moving on to production of cheese. Production of cheese involves three main steps. The first step is curdling, second separation of curd formation, and third one is ripening of cheese. Curdling. Curdling in turn consists of three parts: milk preparation, acidification and coagulation or coagulum formation. Milk coagulation occurs during inoculation with bacterial cultures like Streptococcus lactis, Streptococcus cremoris, usually combined with Lactobacillus lactis and Lactobacillus bulgaricus. Incubation with rennet cleaves casein protein and gives casein hydrolysis. Moving on to the second step, separation of curd from whey. Separation is done by draining, salting and molding. Coagulum is heated at 37 degrees centigrade in order to inactivate the rennet and then cooled. This eliminates the remaining rennet activity and separates whey. Curd is separated from whey, salted and mixed with enzymes. The cheese bricks are pressed to remove excess moisture to enable proper ripening. The third step and the most important step in cheese production is ripening. Ripening involves changes in the chemical and physical properties of the cheese and accompanied by the development of characteristic flavor. During ripening, the curd is gradually digested by the enzymes and the mature cheese acquires the firm or plastic or soft body characteristic of a particular variety, which is followed by preservation of cheese. Let's see a schematic representation of various types of cheese production. Here we can see milk which consists of lactose, fat, proteins and beta carotene are used. And here it is allowed for curd formation, and in this curd formation there are three important factors which are involved. Bacterial cultures the starter cultures produce lactic acid from lactose. It lowers the pH from 4.6 to 4.6 in a period of 6 hours at 31 degrees centigrade. The rennet which is added, it digests the casein, leads to the precipitation of alpha and beta casein at 32 degrees centigrade for a period of 30 minutes. Whereas lipase enzyme, before rennet is added, it will digest the lipids present in the protein. All these three factors will allow for the formation of curd. The coagulum usually consists of lactose, fats, proteins and beta carotene, and the rennet is activated when the temperature is increased to 37 degrees centigrade, and finally The curd will be formed and whey will be the water content of the curd, which is known as whey, will be removed or separated. After separation, the curd is salted, and after salting, again some enzymes like proteases, lipases, producing microorganisms will be inoculated, and during this period protein hydrolysis and lipid hydrolysis will take place. Lipid hydrolysis produces strong flavors due to butyric acid production. Later, the curd is pressed and ripened at 2 to 10 degrees centigrade for 2 to 12 months. Finally, the cheese is produced. This cheese consists of a characteristic texture, flavor, aroma, and a bit of color due to the beta carotenes. This is a diagrammatic representation of industrial production of cheese. The raw milk will be pasteurized, and after pasteurized it is standardized and filtered, wherein coagulation or curdling of milk occurs due to the addition of rennet enzyme, and after curdling of milk the curd will be cutted into pieces, then stirred and cooked further in order to produce the maximum amount of whey, and finally the whey will be drained and this curd will be milled, and after milling it is salted, and this fresh cheese is known as cottage cheese or cream cheese. Apart from this, when it is poured into molds, pressed, draining the any remnants of whey, and when it is ripened, this is known as ripened cheese, which is later preserved at 2 to 12 degrees centigrade. Let us see some of the health benefits and nutritional factors of cheese. Cheese constitutes high concentration of essential nutrients, high quality proteins and calcium. It also contains phosphorus, magnesium, zinc, vitamin A, vitamin B12 and riboflavin. The health benefits of cheese are: it helps in improving the bone health, fights cavity, helps in having a healthy skin, healthy hair, reduces high blood pressure, reduces the chances of obtaining osteoporosis, helps reduce migraine, strengthens immune system, prevents cancer, and helps in producing a good amount of sleep. Next, moving on to the next production is yogurt. A dairy product produced by bacterial fermentation of milk. Common microorganisms used for fermentation are Lactobacillus bulgaricus, Streptococcus thermophilus. Lactose fermenting yeast can also be used. The fermentation of lactose in milk produces lactic acid. The lactic acid acts on milk proteins, giving yogurt its texture and characteristics. How does milk turn into yogurt? Milk sugar known as lactose, which is utilized by lactic acid bacteria to produce lactic acid. The acid causes casein or milk protein to denature and hold water into semi-solid gel, which results in formation of yogurt. Here is a diagrammatic representation which shows the picture of a milk and a yogurt. Milk, we can see the casein protein is mycellus, or there are small bundles which measure about 10 to the power of minus 7 meters in diameter. Whereas in yogurt we can see how these casein bundles, the acidity, lactic acid causes casein bundles to fall apart into separate casein molecules. These rebind to each other in a network that traps the water and produces a gel-like structure or texture to the yogurt. Lactic acid bacteria. Streptococcus thermophilus brings the pH of the milk down to 5.5, allow the milk to clot, whereas Lactobacillus bulgaricus converts lactose to lactic acid. These two bacteria have a mutual stimulating effect on one another. The proteolytic enzymes from Lactobacillus bulgaricus breaks down milk proteins into peptides. These peptides stimulate the growth of Lactobacillus thermophilus or Streptococcus thermophilus, which in turn produce formic acid and carbon dioxide. These are growth stimulants for Lactobacillus bulgaricus. At the end of the incubation, pH may fall to as low as 4.2 to 4.4. The lactic acid coagulates the remaining milk proteins, causing yogurt to thicken. Acetaldehyde, a metabolite byproduct of both the species, give raw yogurt its characteristic flavor. Here we can see how Lactobacillus bulgaricus and Lactobacillus thermophilus, they support each other in production of yogurt. Proteins are broken down into peptides, and these peptides are utilized by Lactobacillus thermophilus. Lactobacillus utilize peptides to form formic acid, acetaldehyde and carbon dioxide. The formic acid, which is produced by Lactobacillus thermophilus, will be utilized by Lactobacillus bulgaricus to produce lactic acid and proteolytic enzyme. These proteolytic enzymes in turn help in breaking down the proteins into peptides. Finally, both the microorganisms produce acetaldehyde as its byproduct, which gives the flavor to the yogurt. Probiotics. In addition to starter culture, other beneficial cultures such as Lactobacillus acidophilus, Lactobacillus subspecies Casei and Bifidobacterium are also present in yogurt. They may be added to the yogurt to improve lactose digestion, gastrointestinal function and stimulate the immune system. Ingredients of yogurt. The main ingredient in yogurt is milk. Type of milk used depends on the type of the yogurt. Whole milk is used for full fat yogurt where the milk solids are about 3.25%. Low fat milk for low fat yogurt where the milk solids are about 2%. Skimmed milk for non fat yogurt where the solids are only about 0.5%. In addition to this, sweeteners can be used, and flavors and fruit preparations are used in yogurt to provide variety to the customers. Let us see some of the major steps used in yogurt preparation. Adjust the milk composition by adding milk solids, then pasteurize the milk and homogenize. After homogenization, the milk is cooled, inoculated with the starter culture and left for a holding period, and after holding period it is cooled and added flavors or fruits, and finally it is packed. Adjusting milk composition to achieve desired fats and solid contents, ingredients such as stabilizers are added at this time. The stabilizers improve firmness and prevent separation of whey. These stabilizers are usually carrageenan, starch, gums, pectins and gelatins, which give the gel-like texture to the, or improvise the gel-like texture to the yogurt, or gives firmness to the yogurt. It is pasteurized at 85 degrees centigrade for 30 minutes or 95 degrees centigrade for 10 minutes. Heat treatment is used to denature the whey proteins. It reduces the number of spoilage organisms in the milk and provides a better environment for the starter culture to grow. Then the milk is homogenized, that is blended at 2000 to 2500 psi to mix all the ingredients thoroughly and improve the yogurt consistency. Homogenization is followed by cooling of the milk. Milk is cooled at 42 degrees centigrade. Cooling is required in order to provide ideal growth temperature for the starter cultures, which is followed by cooling is followed by inoculation with starter culture. Starter culture is important for initiation of fermentation process. The starter culture is added into the cooled milk. The microbes in the culture convert lactose into lactic acid. The milk is held at 42 degrees centigrade until the pH reaches 4.5. The temperature is suitable for starter culture. This process can take several hours. The yogurt is cooled at 7 degrees centigrade. Cooling is necessary to stop the fermentation process. After cooling, flavors are been added, like fruits and flavors can be added depending on the type of the yogurt. Fruits can be added in the bottom of the cup, and inoculated yogurt is poured on the top and fermented. Fruits may also be blended and fermented along with yogurt before packaging. Final step of yogurt production is packaging. Yogurt is pumped into the desired packaging material from the fermentation vat. The packaging material is finally sealed. Yeah, during production of yogurt, we have some microbial problem. The flavor problem can be associated with the concentration of the acetaldehyde. A low concentration of acetaldehyde gives a chalky and sour flavor. Too much acetaldehyde gives a greasy flavor. Too much diacetyls gives a buttery aroma. Too much acid production during storage are associated with bitter flavor. Production of exopolysaccharides by the starters can give a viscous and ropey texture. Growth of yeast during storage can also produce fruity flavor, especially in yogurt containing fruits and nuts. During long storage, molds can grow on the surface. So these are the microbial problems associated with the yogurt production. Thank you.